Good afternoon, everyone. Can everybody hear me? We're about to start pretty soon. Probably in about another minute. I see a lot of people logging in. So those of you who are logged in or are logging in, please read the first slide. We have a lot of people that are online. And so if you can just mute your mic, and if you need to ask a question or interrupt, then you could unmute your mic and we could all hear you. I think all of us can hear each other, and we're going to be starting in a few minutes. We appreciate all of you for joining in. My name is Joe Ng. For those of you who do not know me, I'm here talking to you live, broadcasting from the Hudson Alpha Institute. And here with me is Mark Pusey, who's on my left-hand side. You can see him. And we're here to talk about trace fluorescent labeling and how it can help find early nucleations and ultimately the conditions to find crystal growth. So in terms of where we are at, we have found that we are building a laboratory here at the Hudson Alpha Institute in Huntsville, Alabama. And this is a new biotech institute, and we're working with structural genomics. The trace fluorescent labeling came to us as a method to build faster and more comprehensive methods for early detection screening. So this webinar is sponsored by Express Genes and Molecular Dimensions. And again, we are broadcasting live to everyone out there. And so if you have any questions, you could chat it in, or you could save it until we finish with our presentation. This is about a 15 to 20-minute presentation, so we appreciate your attention. So here we are at the Hudson Alpha Institute. We're actually right here on the third floor, and we have a laboratory that we're working on looking at crystal growth phenomenon. So again, use the chat button to ask any questions. The quality of the sound will vary depending on your internet connection. And after our presentation, you can unmute to ask questions directly. So our agenda really is to talk about what is trace labeling, how is it done, and why did we proceed to develop this, the use of multi-detecting complexes or proteins. And this is actually very important nowadays to look at macromolecular complexes, and in terms of structural genomics for what is trace labeling? The trace labeling is a covalently binding molecules of a small fluorescently emitting molecules, and it's labeled in 1 to 1,000 molecules. It's very, very diluted. It does not affect lattice formation, nor does it affect the subsequent crystallographic analysis. Here we show that a cartoon illustration that a fluorescent molecule can be traced very, very limited. And again, it's about 1 in 1,000 molecules. But you're able to see very intense emission when there is a nucleation event that's ongoing. So Mark's going to explain to you about the basic components of the microscope that's used to apply for this technique.
So the basic components that we're using, these are really true for any fluorescent microscope. We just put it into an instrument that we think is more usable. For a fluorescent microscope, we're using epifluorescence. We have a light source. We're using LEDs. We pass it through an excitation filter, which cuts off the higher wavelengths from the LED. Then that goes into a dichroic mirror, which reflects the light down through a focusing objective onto the sample. The fluorescent light which is emitted is picked up by the same objective, is transmitted through the dichroic mirror, and then goes through an emission filter, which removes the lower wavelength lights, and then goes to a detector. Now, the detector in this case can be a photomultiplier tube, the human eye, or in our case, a video camera. I show this on the side here that we're using visible fluorescence, and the reason for that is that we can use a number of different emission wavelengths so that we can get multiple colors out of this. We use this for multiple colors. So how do we label the protein? And initially this procedure took a long time. most of the better part of an afternoon. We've now worked on it to the point where it can take us less than 30 minutes and as short as 5 minutes if possible, or if needed. So we start off with our canonical end goal of 1 milliliter of protein at our crystallization concentration. And what we actually start off with is 0.8 milliliters of protein at 125% of the desired final concentration. So from that, we remove 0.08 milliliters or 80 microliters. So 1/10 of the volume is removed and buffer exchanged if necessary to put the protein into the reaction buffer. The reaction buffers cannot contain any primary or secondary amines since our procedure is labeling the N-terminal or the side chain amines, and we're doing it randomly. To this, we add, after the buffer exchange, we add 0.5 to 2 microliters of reactive fluorescent probe solution. We use closer to 2 microliters for the higher molecular weight proteins and smaller amounts down to the 0.5 for lower molecular weight proteins. We incubate this solution while we are equilibrating the desalting column into the buffer that the protein had initially come out of. If we want to really speed this up, we use 2 desalting columns, one of them already pre-equilibrated, and this is what gets us down to the 5-minute reaction time. Seem to have lost control here of things. Okay, so we do the final desalting back into the protein buffer, take what passes through from there, and add that back to the original solution, and bring the volume up to there from there to 1 milliliter. So and we have our labeled solution. Now, the solution itself will not be as intensely colored as this. In fact, I'm colorblind, so I usually can't see any color. However, other people have shown me that there is usually, for the probe we're using, a very faint color to it. Anyway, you can see through this procedure that we start off, the name of the game in this is trace. And we start off at the very beginning by limiting the amount of protein that we're using or that we're even going to react, and then we try and minimize the amount of that which is reacted because we really only want from 0.2 to 0.5% of the protein to be labeled. We can go with lower if we have higher sensitivity, but optics. So why are the lead results so much better, and why do we do this? Well, it's very easy to see when we're fluorescently labeled our results. And this extends not only from the crystalline results, which you'll see later, but even hidden leads, which you didn't know that you have. And here's a case where we have a precipitate under white light. There's nothing there. Most of us, if we just saw this on our well, would dismiss it and as a crystallization condition. However, when we look at this under fluorescent conditions, we see that there are bright spots all through here, which do not correspond to any structures that we see up in the white light or anything structural. But the general rule of thumb is for fluorescent trace labeling that intensity equals structure. The more dense the structure is, the more intense the illumination will be, or the fluorescence emission. And the crystal being the most dense structure, they're going to be the brightest of the emitters. So we took these conditions for this protein, which I believe is beta-lactoglobulin, and we optimized on those conditions, and we successfully obtained crystals. So this is one of the advantages of this, is that we get these extra conditions that we don't even see, and we expand our leads in all of this. So there's no chance of finding salts for this. One of the initial reasons for using fluorescence is that the salts themselves will not be fluorescing. So if we get these promising looking results of salt crystals under white light, and we put them under fluorescence, and we see that we get, instead, we don't see any fluorescence. In fact, what we see is a shadow or a black hole in there corresponding to the salts. So we know that these are not protein crystals. Precipitate is not a problem with this. Looking for straight lines in general, if you're doing a white light only scan, can be difficult. However, especially if we have precipitate present, And here we have protein crystals which are buried in a precipitate or in a precipitate. If you're rapidly scanning through your plate, you may or may not see these. However, when we go into fluorescence mode, we see crystals, the light from the crystals shining through, much like light bulbs under the snow. It's very easy to see that we have protein crystals there, although you may not see them on a cursory examination of the white light only images. We have empirically determined that we can see crystals at least to the size of 10 microns, maybe smaller. This is more a function of the optics than of the use of fluorescence itself. We're using relatively low magnification optics, but we can still detect and see crystals smaller than 10 microns with what we're using. It's easy to review the results. Here is an optimistic or a very happy outcome where we're looking at the images from a screen, and we can see that, again, with intensity equaling structure, we can step way back from here, and we can see all the conditions of interest in this. They shine through. They're very easy to see. We can step back even further and see them. We don't have to be up close or ruining our eyes looking through a microscope to find these. It's easy to see progress, and here we have an outcome where we had a day 1, there was nothing. By day 3, we had what was apparently a spheroid under white light, and we see that by day 18, the structure had grown, indicating that it was possibly crystalline. In fact, by day 18, we're also seeing the emergence of facets on this, over here and over here, and eventually this became a faceted crystal. But we can see this, and we can see where our interesting conditions are. We can use more than one emission color, which I alluded to earlier on. In this case, I took the same protein and labeled 3 different populations of it with 3 different fluorescent probes, one emitting blue, one emitting green, and the other one emitting orange. Then mixed them back together again and looked at each of them, looked at each population under the appropriate fluorescent conditions using the appropriate excitation, wavelength, and filters. Excuse me. We also did controls where we emitted at least one of these colors from the different populations, and we found that we could not or would not see, when the probe was absent, we did not see the fluorescence from it, and so we can use this now as a means of testing for the presence of different labeled subunits in a complex. So, back to Joe.
So, as a crystallographer, one would always ask, why would I want to put in some trace contaminant? And as we said earlier on, this is actually a very, very small amount, thus a trace labeling. So it's about 1 in 1,000 stoichiometric ratios. And what we wanted to show you is that the trace labeling does not affect crystal quality. So we're going to show you just an example of an X-ray diffraction pattern. Now, listen to me carefully. This is not lysozyme. Everyone's showing model proteins. This is not canaverin or thaumatin. This is actually one of our structural genomics protein. This happens to be the inorganic part of phosphatase that we've been studying for a number of reasons, for X-ray and neutron. And we've gone through a lot of searches. But this is just an example of a diffraction pattern that is taken at a home source. And this is a very short exposure. It's about 5 minutes. So it's this crystal normally diffracts very well, but you see it cuts off at about 2 angstroms or so. But it's just to illustrate that it does give a very, very low mosaicity diffraction pattern, and this gave out a complete data set which we were able to solve the structures of these. So again, it does not affect, at least not in our hands, the ultimate crystallographic structure, nor the means of interfering of how to, of the data interfering with it. So why did we build a CrystalX2? And as I mentioned before, our structural genomics program, a number of years ago, we were obligated to look at hundreds of crystallization screening conditions. We had a genome relatively small, but we had 2,000 proteins to look at. And so we're cloning and expressing, and now we're really looking at trying to find early leads for hundreds at a time. So it's no longer it's a very agonizing procedure, as all of you know, we've gone through a number of proteins. So imagine going through hundreds of these. And we're a small lab, so we don't have any fancy automated machines other than students with a lot of multi-channel pipettement. So this is the motivation to build this instrument, was to facilitate mainly for us in the beginning that we could find early leads very quickly and efficiently. And then thereafter, when we actually built the instrument, we thought that this could be usable for the X-ray crystallographic community. So in terms of screening, storage, and research, and this is actually using LED, not UV, and so it puts an extra safety margin on us because I did work at I'm still working at a university, and it's actually important to not have UVs, mainly for the safety of students and to meet all safety standards. So that's the motivation behind this, and the efficiency it takes about 4 seconds to look at from camera to screen. So again, when we're looking at hundreds of these, this becomes much more efficient. So in a 96-well plate, we could look through and scan the entire plate in about less than 15 minutes. So it's pretty fast. So this type of automation can be archived as well too. So you can have hundreds of pictures that you go back and look at and evaluate. So here again, we're going to show you again live broadbanding. So we're going to move the camera a little bit, so don't get car sick. So this is in your screen, you should see an icon with 3 people on there. And so if you could click on that, it will expand your screen so you could actually see the instrument very well. So again, this is live broadcasting. We're going to move the camera a little bit and show you really quickly what this machine looks like. And so again, our motivation driver was to try to look at crystallization plates. Well, actually, there's 2,000 of these plates, so it's a 284 so crystallization screen that we look through. And so we're going to put this on this tray here, and each one is set up with different conditions, and you could use multiple different types of screening solutions. And this has a fluorescent label on the proteins. So if, Mark, you could turn on the instrument, this actually shows you what it starts out looking for, and it'll show you a white light capture, and then followed by a green light, and then these excitation wavelengths will allow you to look at different types of single crystals that may arise very early on that you would not normally detect otherwise from a light microscope. So here you see the yellow light, the green light, and we're looking at different complexes. You can see the white light. So you have all different pictures to compare it with. So this is our concept. This is an automated machine that allows us to screen things very, very rapidly. And as I said before, that we could actually screen this entire tray in very, very rapid time in about 15 minutes or less. And if you go for higher plates, these will take a little bit longer, but as you can see, this used to take us all day, if not all week, to look at this. And from scrutinizing over, viewing the microscope, and hunching over, it is quite agonizing in the beginning, so this has facilitated a lot of the procedures for us. So Mark's going to go over what you see in the software, and this is actually a very easy software, and Mark will take you through it.
Okay, so the CrystalX2 software was designed for simplicity, and we're going to elaborate on it eventually, but it is currently made to be very simple. So this is what the basic experimental setup screen, which we get by going into setup from the main screen. We created an experiment, which you can put the experiment name in here, and this would be for each plate that we're using, and then the plate type. And we can add just about any SDS standard plate to this. We have gone from the standard 96 well plates up to 1536 well plates that we've been able to add to this instrument. Down here, in this section, we can edit these parameters for each different white light or light color source. And what we're adjusting down here are the camera parameters for the exposure time, the rate at which the frames are taken, the gains for the different colors of the camera and the master gain, plus the Z position, since different light colors are going to offset or focus at different points, and we can adjust for that here as well. And the other main screen, not much to see on this, is by going into the mode switch, we have the auto mode, which is what we normally collect our data with, manual mode, which we use for, as it says, manually adjusting the position and the focus and the intensity, light intensity. The home mode, this basically homes the stages and only has to be done whenever the box itself has been turned off or turned on, one time only, and then exit, which should be fairly self-explanatory. So we have now another demonstration of the instrument in operation, and here we go when we launch the software. And this is the basic data collection screen, and here we can select the well that we're going to start from. We're starting, in this case, from well D1A on this plate, and it is now moving to there, and then it just starts acquiring images. We're just using the green in this case, and here is D1A, B, and C, and then we can go into specific wells and blow them up while we're collecting the data and to see what we have in those wells. Do that again, blow it up, and again you'll notice that we're still collecting data as we're doing it. These are rod-shaped crystals. I can tell that because we're getting higher fluorescence on the ends. It's a light pipe effect that we get from rod-shaped crystals. And here are the last one that we're blowing up on. And then we're not going to see this last one because that ends this demonstration. Okay, so the benefits of the trace labeling. And this, we're back to Joey.
So as I pointed out, the motivation, the driver behind this was really to go through hundreds of these proteins. So it does speed up your screening process. So the goal is to try to make the research easier if you're into high throughput, high output mode. But the more important part is scrutinizing over the microscope. You know, it's very, very difficult, and everybody has a different quality check. You know, what seems to be a early nucleation to someone may not be otherwise. So it's easier to find leads, and it's more consistent, and it's easier to see and monitor the progress. If you're looking at hundreds at a time and have comparison, have a reference picture to look at, then it's quite much more easier. And as I pointed out before, if you're looking at multiples of proteins coming together, you're talking about a dimer, a trimer, you could easily hook these, covalently bind these proteins with the fluorescent molecules. And if complexes are formed, then you could see the differences in color. Whereas before, you wouldn't know really if you have a complex or not until you undergo an X-ray diffraction analysis, which does take more time. So the trace labeling, arguably, said it's also good for your health too, as I mentioned before. It does relieve eye strains and gets rid of all the headaches and stress and all that stressful stuff that comes with it. So anyway, you get a happier lab, I think. It certainly made our job easier, but on a serious note, I think the science behind it is very meaningful in terms of finding early leads, finding early nucleation. It could be a method to really study early nucleation approaches. So at this point, you're gonna turn on the I'm gonna put the camera on to me and Mark and me, and we're gonna entertain any questions if you like. Either turn on your mic or you could chat it in, and we'll be happy to answer any questions. So first of all, we had we don't know if the questions are coming in chat, so there's a number of shy people who are shy out there who don't want to speak up. So the first question is coming in, if I could read this far off the screen, and I think says that is there a supply list for the trace labeling reagents? And the answer is yes, there are a number of probes out there. Molecular Dimensions has put a trial kit together that is available for anyone of the asking. So if you could contact Molecular Dimensions or contact us, and we could certainly email you. The question has just come in. Are other labs using the CrystalX2? Yes, we have users both in the UK and the US at the moment, and so far they've been happy with it. There are a few more chats coming in. Feel free to open up your mic and ask any questions, and we're here. And okay, we do have another one. Does this work with membrane proteins? And yes, we've talked about complexes, and this worked in the presence of detergents as well too. It works for optimization for procedures. We're undergoing a study using LCP at the moment, and Mark is looking at a number of membrane proteins for that.
The difference with using membrane proteins is that the labeling procedure needs to be modified to get rid of the free probe, to make sure that you don't have probe lurking in your vesicles or your micelles.
Sorry, I think there's a voice coming in, and I'm sorry I can't hear you that well. We do have a few more chat questions, and I'm trying to listen in to see if anyone is trying to ask any questions here. Well, we have another chat question. Can you see the probe in the electron density map? Well, that's a very good question. That's a very common question. As I mentioned before, the ratio is so low, and when the structure is solved, you just do not see any electron density for the probe. We've done it many times. We've solved structures directly. Just to clarify, when you label these proteins, the early conditions is not necessarily the protein crystal that you can use for structure determination, even though often the case it is big enough to shoot directly. You can do that, but its originally intended purpose is to find the conditions, and then you optimize it with the protein that has and does not have a probe on it. And usually it is pretty consistent in terms of having probes or no probes. So no, it doesn't show up in the electron density map. It doesn't show up in the ultimate structure determination, and the probe is pretty much invisible. Okay, someone asked me to go to the last slide, and I'm just reading the chats that's coming in. So a chat question came in and asked if there are published references for this, and yes, there are. I just put up a slide up of the publications that is either submitted or published already. If you want to learn more, contact www.moleculardimensions.com or just go to the website of www.crystalx2.com. And at this moment, let me thank them who have participated in this webinar, and we'll stay on just for a few minutes for anyone who wants to answer any questions answer any questions. I'm sorry, the audio may not come across. If you had tried to do an audio with coming in, maybe you could try again, and we could listen carefully. But for the remaining of you, thank you very much again, and we'll stick around for any questions.
Hello, this is Herman. I have a question. Are random amino acids labeled or specific amino acids being labeled?
The question is the label random, or can you specifically label to a specific location?
If you have a particular location that you want to label, you can probably target it. However, the way we're doing it right now is we're randomly labeling side chain amines. So we put our labeling pH of our reaction up around 8.5 to 9, We put it down at lower pHs, around 7.5. So targeting the N-terminal amine, we'd rather have a random labeling than a specific labeling, just to further avoid any other complications.
Okay.
Okay, Rob, there's a chat question that just came in, and sorry, it just popped in. I couldn't read it. Can you chat it again, please? So the question is, do you have any problems with the label component of a complex aggregating on surface of a subcomplex crystal lacking component? So do we have any problems labeling components of a complex aggregating on surfaces of subcomplex crystals? So we've tried very limited numbers of different types of surfaces and different types of crystallization chamber. We haven't run into any problems yet in terms of aggregations on the surfaces or any of the components thereafter. It's due because of the very, very limited number of labeling molecules that's on the protein. So I'm getting a lot of statics, and there's a lot of questions coming in. If you're not talking, could you please just mute your mic temporarily? Otherwise, we're delighted to listen to your questions. It's not coming in too clearly. Come in is, where can I obtain the kits? Again, there are kits now available containing the fluorescent probe that you can actually try out, and Molecular Dimensions is distributing that. So you could contact your Molecular Dimensions distributor for any type of free trial kits that you could use. Okay. All right. So we got. Okay. So we have another chat question. It just showed up. So all of a sudden the traffic became very, very high. Do you have a problem with the label component of a complex? No, sorry, that was asked already. How does CrystalX2 compares to a current alternative technology, and what are the advantages? Well, first of all, a comparable technology may be to use UV fluorescent. Use UV illumination, and using a fluorescence here, we've had no incidents so far where we had false positive. And I think with the equivalent using UV illumination, there are still instances, at least in my case, that I see non-protein crystals come out, either part of a detergent for membrane proteins or salts that still illuminates under UV. If I compare it with the fluorescence, and I'm saying this on a one-on-one because we did look at this, we see that it's more consistent in results. And as I mentioned earlier in the talk, it is safer and it's much more simpler because you're not dealing with an ultraviolet radiation. And so if you're in a laboratory where safety is concerned and that is a limiting step, I think the LED becomes much more advantageous. So we got a couple more questions coming in, and I'm just trying to read these chats. We got a number of typings that are coming in. Question is, how big is the fluorescent probe, and what's the size that you're allowed to use fluorescent probe without interfering with the crystallization?
When we were initially working this out, I used the same fluorescent probe, which has a molecular weight of 550, and I used it with insulin crystals. So there the probe was about 1/10 the size of the protein molecule itself, and we had no problem with the diffraction pattern or crystallization procedure. especially at the trace leveling levels.
Well, if there are no other questions, again, thank you very much, and thank you for all those who are staying on and asking all these great questions. Feel free to contact Mark or myself. We're on the webpage directions that you've seen on the slides, and also you could contact Molecular Dimensions as well if you have any more questions. All right, thank you, all of you. Good night, good morning, good afternoon.